Hello everyone, this is Tejaswi Dinakaran. I will be talking today about linked read sequencing. Linked read sequencing is a type of DNA sequencing technology that uses specialized technique that tags DNA molecules with unique barcodes before fragmenting them. Unlike traditional sequencing technology where DNA is broken into small fragments and then sequenced individually, resulting in short read lengths that has difficulties in accurately reconstructing the original DNA sequence, the unique barcodes of linked read sequencing allows the scientists to link together DNA fragments that comes from the same DNA molecule. A pivotal benefit of this technology lies in the small quantities of DNA required for large genome information output, effectively combining the advantages of long read and short read technologies. Steps of linked read sequencing are as follows. Sample preparation. The DNA is extracted from a sample and cut into fragments of 50 to 200 kilobase pair long. Second is barcode sequencing, where each DNA fragment is labeled with a unique barcode through a process known as gel bead in emulsion, which is also known as GEM. Library preparation. Library preparation includes barcoding of DNA fragments which are then amplified with PCR to generate sequencing libraries. Sequencing is done with the help of Illumina next generation sequencing technology which generates millions to billions of short sequences reads that represents fragments of the original DNA molecules. In barcode processing, grouping is done of short reads to longer fragments based on the barcodes. Downstream analysis includes processed reads which are aligned to a reference genome or used for de novo assembly of complex genomes, haplotype phasing, or identification of structural variations. Now let's talk in detail about the linked read sequencing steps. Barcode sequencing. During barcode sequencing, high molecular weight DNA samples that contain the target DNA sequence, ranging from 50 to several hundred kilobases in size, are combined with the gel beads containing unique barcodes, enzymes, and sequencing reagents. Microfluidic device can partition input DNA molecules into individual nanoliter sized droplets of water in oil emulsion known as gems. Each gems contains gel beads coated with the same barcode and primers and a small amount of DNA. The primers are complementary to specific regions of the DNA molecule, allowing for amplification of the DNA in the droplets through PCR. The barcodes enable the identification and grouping of sequencing reads that originate from the same long fragment, which is crucial for downstream analysis. Library preparation and sequencing. The barcoded DNA fragments are amplified using PCR to create a library of DNA fragments with identical barcodes. All the fragments derived from a given DNA molecule are tagged with same barcode. This step increases the quantity of DNA for sequencing and reduces these chances of losing unique DNA fragments during sequencing. Droplets are collected in a tube and the emulsion is broken, releasing the amplified barcode DNA sequences. Standard Illumina next generation sequencing technology can be used to sequence the libraries. During sequencing, the barcodes are read along with the DNA sequences, allowing the researchers and scientists to group together DNA fragments that originate from the same DNA molecule. Even though each DNA fragment is typically not fully sequenced, the information from many overlapping fragments in the same genomic region can be combined to reconstruct the original stretches of the genome. Therefore, a genome can be easily assembled from scratch without any prior reference. Processing. The raw processing data is then processed through bioinformatics tool to remove low quality reads and to assign reads to their respective barcodes. Reads can be aligned to a reference genome or assembled de novo to generate long rich contexts. The read alignment step is important for determining the order and orientation of the long DNA fragments and for identifying genomic variations such as insertions or deletions. Now let's talk in detail about the Illumina next generation sequencing, which is part of linked read sequencing. So, during library preparation, the genomic DNA is fragmented into small fragments, which is shown in the first step. Then the specialized adapters are attached to both ends of each segment through ligation, which is shown in second step. The library is then loaded into a flow cell or chip and is amplified into a cloner cluster through bridge amplification. First, the DNA segment is amplified through PCR, which is shown in the first step. Then the original segment is washed away, which is shown in the second step. Then the segment's bends attaching itself to the adjacent adapters present on the chip. This bridge is then amplified using PCR and then denatured. In this way, the bridge amplification may continue, creating a cluster of clones in the process. Before the sequencing can start, the reverse strand is cleaved, which is shown in the following slide. Now, these different clonal clusters can be sequenced through synthesis. Can be sequenced through synthesis. Now, let's examine the process in detail by only focusing on a single segment. Sequencing reagents are added, including nucleotides, which are fluorescently labeled, meaning that we can detect the nucleotide when it attaches to the segment. This can be done by due to the wavelength and intensity emitted by each of the fluorophores attached to the nucleotides when we shine a laser on them. This is done simultaneously for all clonal clusters to create individual reads. Now we can align these generated reads to a reference genome with bioinformatics software. After alignment, any differences between the newly sequenced reads and the reference genome can be identified. Now let's move on to the advantages. The advantages are as follows. Accuracy and scalability. Method requires small quantities of input DNA, which can be beneficial for small samples or single cell studies. More cost effective. Library produced by Linkread can be processed using Illumina short read sequencing, which increases the accessibility. Next is disadvantages. The complexity of library construction. This technology requires high molecular DNA preparation in order to produce long enough DNA molecules for sequencing. Limitation in read length may result in limited haplotype resolution, which could reduce the efficacy of this technology in highly complex genomic regions. Applications. First is de novo genome assembly. Linked read sequencing can facilitate de novo genomes assembly, which involves reconstructing a genome from scratch without any prior reference. Linked read sequencing enables assembly of large genome regions and helps to improve the completeness and contiguity of the resultant genome. This can be particularly useful for studying organisms that lacks a high quality reference genome, such as non-model organism or organism with complex genomes. Haplotype phasing. Haplotype refers to a group of genetic variants inherited together on a chromosome from one parent due to their genetic linkage. Haplotype phasing refers to the process of reconstructing individual haplotypes important for determining the genetic basis of disease. Linked read sequencing allows consistent coverage of genes related to different diseases, helping scientists to obtain all the regions carrying mutations from targeted genes. Structural variation analysis. Structural variations such as deletions, duplication, inversions, translocation, and other rearrangements are common in human genomes. These variations can have significant impacts on genome functions and have been implicated in many diseases. Linked read sequencing technology labels all reads that originate from the same long DNA fragment with same barcode, so it enables the detection of a large number of structural variants. These are the references